All right, I guess we will get started with I'll let you sit down. The last session before the fireside chat. Okay, so for this session, unfortunately, we will have two speakers instead of three. Ervin Asokan is not able to join us today, but I think I'll be giving the first talk, and then Alison Keeler will be giving a talk that I'll introduce in a second. So thank you for your attention. The other thing I'll mention, I think we're going to hold questions out of interest and time until the end of both of our talks. Okay, so I'm going to talk today about my lab's approach. So for those of you who don't know me, I'm Ben Deverman. I work at the Broad Institute here, just upstairs, and my lab develops AAV capsids for delivery into the CNS. And so we are focused almost exclusively on trying to use the vasculature to disseminate our capsids throughout the CNS, and we think this is really critical because it's really only through the vasculature that we can reach all regions of the CNS quite well. But of course, as we've heard about today, this is associated with a number of challenges. One is that the natural AAVs are not that efficient at crossing the blood-brain barrier and reaching the CNS. When there has been a lot of work in the last few years from our lab and many others that have shown that you can engineer this problem. We can fix this through capsid engineering and develop capsids that cross the BBB in animals. And the challenge there has been really that this has been, in a lot of cases, quite species specific, or at least limited or restricted to certain species. And so this has been a challenge, but I think there are a lot of really encouraging signs that some of these capsid engineering efforts have developed capsids that are going to work in humans. And so the proof will be in the next few years. And finally, the thing that we're all here to discuss is that the other challenge is, of course, pre-existing immunity. And so there's a significant fraction of patients that would be excluded from clinical trials or getting approved products due to pre-existing antibodies. And so today, I want to tell you about our work to try to address both of these problems. But first, I just wanted to mention that our goal here is really real. Even here at the Broad, we are trying to work to develop a gene therapy for prion disease. And so we are doing this in collaboration with a really great team here. This project's led by Sonia Valab and Eric Minical. Eric is in the second row there. So Sonia is herself a carrier of a mutation that will cause prion disease, and we are working with their team and David Liu and Jonathan Weissman's lab, who are developing cargos as ways of turning off prion disease to try to develop a therapy. And this is really puts this in real terms that we need to develop capsids that can deliver genes throughout the entire brain to neurons throughout the entire brain to make this successful. And of course, we want to do this in as many patients as possible and address the neutralizing antibody problem. And so really what we're trying to do in my lab is first develop capsids that can cross the BBB in humans. We've done this in mice, we've done this in non-human primates, but can we get it to work in humans? And second, we want to build in antibody evading capabilities into these capsids. And so today I'll tell you about both of these efforts. So to get AAVs that would cross the blood-brain barrier, we've been focusing on developing capsids that are targeting specific human receptors as a way of addressing this problem with the species barrier. So we are going directly after human receptors. And the first receptor that we've targeted is transferrin receptor. Transferrin receptor we think is a great choice for a couple reasons. One, it's highly expressed on the human blood-brain barrier. It's a well-characterized mediator of transcytosis across the blood-brain barrier. And in fact, there are now at least 3 companies that have demonstrated target engagement in the brain with transferrin receptor binding biologics, so usually antibody-based therapies. And so this is really exciting. This is now a validated target in humans, so that gives us a fair amount of confidence that what we're developing in mouse studies that I'll show you could actually work in humans. Okay, so we've developed a capsid called BI-HDFR1 that can cross the BBB by using human transferrin receptor. And This work was published earlier this year, so I'll go through just a few highlights from it. But basically, we showed that we can deliver AAVs throughout the brain of humanized mice that have humanized transferrin receptor knocked in. And these same AAVs, when you deliver them to wild-type mice, do almost nothing. They look actually worse than AV9. They don't cross the BBB hardly at all. And they're going into both the brain and spinal cord. And we see pretty high levels of neuron transduction across multiple regions of the brain, as I'm showing on the left. In the pink bars, those are the novel capsid in the transferrin receptor knock-in mice. And as you can see in the wild-type mice and in the color that you can't actually see, there's really no transduction whatsoever. So since the publication of this paper back in May of this year, my team has been working really hard to try to develop even better versions of this capsid. So we've generated second-generation versions through screening in the transferrin receptor knock-in animals. And this is just some early data from some of this screen, where we've dosed these animals at an even 10-fold lower dose. So this is animals dosed at 2 × 10 to the 12th vector genomes per kilogram. So this is roughly 50-fold lower than the doses that are currently used in DMD gene therapies and for Zolgensma for SMA. And we're still seeing really broad distribution across the CNS. The first one here on this panel is our first-generation capsid. This is our second-generation capsid. And then as a comparison, this is comparing it to PHP-EB, which is still a standard capsid used as a surrogate in many mouse model studies. Okay, so our next challenge is to try to address the pre-existing antibody neutralization to try to expand the treatable patient population. And so we're doing this with our little gold capsids. Okay, so what we did to try to address this problem is we started with our second-generation transferrin receptor targeted capsid, and we made a bunch of additional surface modifications in this capsid to try to get it to evade neutralizing antibodies. And so we made a library of many hundreds of thousands of different variants from this approach, and then we screened them for their ability to resist neutralizing antibodies, for their ability to continue to use transferrin receptor to get into the CNS, and then to have maintained manufacturability and also to be compatible during manufacture and purification with commonly used affinity resins for purifying the AAVs. And so we then selected a few capsids that I'll show you. But first, when we wanted to test these to see whether they were actually working, what we had to do first is really establish a whole set of human sera that we could test these capsids against. And so we have taken over 60 human sera samples now. These are ones from the US, but we're building this and also getting samples now from other places around the world. And so we've taken these and looked for neutralizing antibodies against AV9 as our first pass screen. So what I'm showing you here are the IC50 or half inhibitory titers against AV9. We're seeing about, in this set, there were 46% of the samples were neutralizing to AV9 at some level. The ones on the bottom are the ones that show neutralizing titers. Some of them are, on the moderate range, as others have shown, and some are very strong. And this is with a neutralizing assay done in HEK cells with a very low amount of AAV, so that this is a very sensitive assay, at least in our estimation. And we then took our capsids, and from our library we picked over 50 capsids to test to see how well they evaded neutralizing antibodies, and we screened them against, as a first step, just the 8 least neutralizing sera. So not the ones with no neutralization, but the ones that had the least neutralization. And so this heat map that I'm showing on the right is basically what you're looking at is each column is an individual AAV capsid, and each row is a different human sera. And the colors in the heat map, if it reaches yellow, that means the capsid is showing activity in this assay. So it's not being neutralized, and the ones in blue are kind of partially neutralized, and if it's purple, it's completely neutralized. Our starting capsids are these. There's a couple preps here on towards the left, so these are ranked by their average neutralization. So their starting capsid is pretty much completely neutralized in almost all the assays, and we've developed a bunch of capsids that have some activity in the presence of these human sera And in this case, we're not running full curves. We are just looking at a 1-to-4 dilution of the sera as a first pass test. So once we did this, we picked 5 variants that seemed to be the best at evading neutralizing antibodies. And we tested these against a full panel of neutralizing sera that I'm showing in the same assay. So this is just a single neutralization amount, 1-to-4 dilution of the serum. And we see that we have some of the capsids are evading neutralization completely in this assay to the detection limits of this assay. So this is really encouraging, but we still have a long way to go. One of the questions that we then had, of course, is do these still function? And so from our library data, it appeared that they did, but we wanted to test them individually. So these are low-dose injections into the transferrin receptor knock-in animals. This is our second generation capsid, and these are the 5 capsids that have better antibody evading abilities. And you can see that all of them still cross the BBB and transduce cells throughout the brain with similar efficiencies as the starting capsid. So we then, from that pool of 5, we now narrowed it down to 1. And so all of this data here is with our best performing antibody evading capsid. And so I'm going to walk you through a little bit more in more detail about this. The graphs on the top are showing the full neutralization curves that we did with increasing sera and how a few of these performed. So, and the heat map on the bottom, this is actually IC50 data, not just data from a single 1-to-4 dilution. This is actually full dilution curves. And so anything in white is non-detected, no neutralization detected. And as you go to darker gray, then those are ones that are highly neutralized. And just as a few examples here, this serum number 2, this is what the neutralization curves look like. We saw no reduction of activity when we added increasing amounts of sera. For this human sera, this number 28, which is here, we see that our engineered capsid that evades antibodies better is able to keep full activity in this assay, while AV9, AV5, and our parental capsid are fully neutralized down to the 1-to-4 dilution. And finally, one more example in this human sera 40, which is the most neutralizing sera sample we have. This one, we were not able to overcome the neutralization, but we did shift the curve by over a log. So still making really good progress. And as I think we've talked about, it's really hard to know exactly how these in vitro neutralization assays will relate to the in vivo situation. But this patient would probably still be one you choose not to dose. But still, I think we've made pretty good progress, and you can see there's a bunch of patients in this range that may potentially be dosable. Or I shouldn't say patients, these are individual human sera, not patients, but based on this in vitro neutralization assay, this looks very promising. And again, this is an ongoing study that we're working on to more carefully assess this top capsid that we have versus our second generation capsid and a few other references where we are doing a 6-animal study across each of these and getting better biodistribution data and quantification data that we'll have shortly. So one last thing that I wanted to mention is that we really built this from the beginning, and this credit goes to Simone Pecqueray, who's in the lab, who's really led this project. We really built this from the beginning we hoped we could make it a modular platform. We didn't want to just solve our problems with our capsid. We actually wanted to be able to take these antibody-evading mutations that we're making and combine them with other tropism-modifying peptide insertions that other groups had developed. And so to test this possibility, we used a capsid called one of the MyoAV capsids developed by Sharif, who's somewhere in the room here, up there in the back, in Party Sabeti's lab here at the Broad. This is a capsid that has a RGD sequence in it that is using an integrin to get into muscle and heart very effectively. So we asked the question, could we take one of these peptides that have been developed by another group and not screened in our library and use one of these capsids that we've developed to make it more antibody evading? And so the answer is yes, we've made some progress on this. So this is the same type of heat map I showed before, where we're testing the capsids against a single sera concentration. This is from our same set of neutralizing sera that fully neutralize AV9. MyoAV is also fully neutralized, as one would expect. Typical loop 8 modifications in the capsids don't usually evade antibodies much better than the parental capsids. But then once we've moved in our antibody evading mutations, we see that we've made significant progress on being able to potentially treat individuals who have neutralizing antibodies to some of these parental capsids. So do they still work? The short answer is not all of them. What we've built here is a platform technology that I don't think there's a single right antibody evading capsid set of mutations that are going to work with everything. But we did find that this one antibody evading capsid here maintained full activity of the MyoAV capsid in the mouse in both muscle transduction and heart transduction, and also the slight decrease in liver tropism that was seen with the parental capsid as well. Okay, so a few, just to summarize our findings so far. Our second generation transferrin receptor binding capsid is able to efficiently transduce the CNS, and this uses a clinically validated mechanism of action. So we're really hopeful that this is going to work in humans. We've identified transferrin receptor binding capsids that evade a significant fraction of neutralizing antibodies in human sera, and even ones that neutralize even AAV5. So our data so far is that these capsids are actually more antibody evading than AAV5, which we think is pretty exciting. And we built these as a suite of antibody evading capsids that we think can now be combined with tropism modified capsids from our group and others. And so questions and next steps. I mean, a lot of these that I put in, I knew would be naive questions that would be asked many times during the day, but we can still go through them one more time. As Juliette Hordeau mentioned, repetition is great for our memories. So you know, some of the ones that we think about a lot are how well do the in vitro neutralization titers align with neutralization in vivo. We still need to show this for our capsids. All the data I've shown you so far is data that is in vitro neutralization assays. We need to really study these a lot more and understand how they react to total antibody assays, and also how well that's going to translate to in vivo neutralization. We've also talked about what happens when you dose patients who have pre-existing immunity and whether they're going to be more susceptible to T-cell mediated cytotoxic effects, or maybe even CD4 T-cell effects. And we've made some progress. We don't think we've engineered all the parts of the capsid that we really need to evade all antibodies. So, you know, we plan to do at least one more round of screening to try to identify capsids that are even better at evading neutralizing antibodies. And the question is, how close can we get to being able to dose almost anyone if we keep this going? And finally, I think the holy grail of this would be to be able to engineer capsids that are far enough away from each other to be able to redose potentially, that all still had a common mechanism of enhanced tropism, but were so different from each other that they could potentially be redosed. And of course, all of this is probably not going to be possible by capsid engineering alone, but I think hearing that there's been so much progress with immune-modulatory strategies, like combining these two could be really powerful. So with that, I'll just end and thank everybody on my team that has done all this amazing work. And I think a lot of this work's really been led by Simone Pacqueret and Ken Chan, and gotten help from a tremendous number of people on my team. And our Prion project is, you know, as I mentioned, is really exciting and giving us strong motivation to make all of this work. So I'll stop, and I won't take questions now unless we're really doing well on time, but I imagine we're not. So I will, I'll turn things over now to Alison Keeler. So Alison is an assistant professor at UMass in Worcester, and she's an immunologist who has expertise in T cells. I think she's going to tell us today about what is known and what is not known about how T cells or how pre-existing immunity may affect the T cell response, and also talk about some of the exciting work that she's doing trying to develop CAR T therapies to help AAV therapies work. So thank you. I'll turn it over to you, Alison. Just grab this. That's okay.
Thank you for the kind introduction, and thank you for allowing me to come and present here. It's really an honor to share the stage with many really important people in the field doing really interesting research. And so we've heard a lot today, as certainly being the last scientific talk about the immune response to AAV gene therapy, so I'm not going to go into too much detail. However, a lot of what we have heard about has really been focused on the B cell response and a little bit maybe about T cell help for the B cell response, but not as much on either cytotoxic T cells, or really at all have we heard about T regulatory cells, or the cells that are really important for immune tolerance. And so there have been a number of studies that have shown that cellular immune responses can be an issue in gene therapy. We've seen that cytotoxic T cells can kill infected cells, and that can lead to elevated transaminases as well as hepatotoxicity. However, in other trials, we have seen that if you can induce T regulatory cells, or these tolerogenic T cells, that you can, these trials have been associated with long-term stable transgene expression. So just to go a little bit more into the data, I actually almost always use one of the early papers, which was shown many times today, concentrating on that there wasn't pre-existing immunity, but there was a T cell response, which wasn't pointed out. But I just want to note in this hemophilia trial, at the same time that there's a peak in ALT, it corresponds to an ELISpot peak. So these are T cells that are specific for the AAV capsid that are being stimulated to produce interferon gamma. However, at the similar time that some of these trials were going on, there was a trial done for alpha-1 antitrypsin, which showed that over 3 years with no immunosuppression, and I should note on the trial that I just showed, they actually gave corticosteroids, and that helped bring down the T cell response. In this trial, there was no immunosuppression given, but there was long-term stable transgene expression. And when the patients were biopsied, they did see that there was cellular infiltration into the muscle. However, what they found is that although there were CD8 T cells, there were also a proportion of Tregs. In the AAT clinical trial and also in the Glybera clinical trial, they also saw infiltration of T regulatory cells into the muscle of these patients. And interestingly, it was Roberto brought up that two of these patients actually had pre-existing immunity prior to delivery, but again, this was an intramuscular delivery, so it has very different immune response. And so when I was thinking about putting to this presentation and coming here, is kind of what do we know about cellular immunity when we're talking about pre-existing immunity? So many people are focused on overcoming NAbs or neutralizing antibodies because they'll prevent, right, AAV from being able to express. But I think it's important, we've heard many, many talks about potential strategies to overcome that, but are they also going to have effect on the T cells? And so really, we regularly are screening for pre-existing immunity, but we're not generally screening patients for cellular immunity prior to redosing. There's also been a lot of talk today about patients who are truly naive versus patients who are potentially negative for, like in antibody assays that we use to screen. And so in my lab, actually, we regularly run these interferon gamma ELISpot assays, but we never do them as pre-screening. They're never used as exclusion criteria. And so it's just something that we potentially should think about. And I know earlier there was a discussion about how B cells and T cell pre-existing immunity correspond. And I think Claudia brought up that in her assays, that when she did TNF alpha ELISpot assays, that there was a correlation with pre-existing immunity. But there was a recent paper published out of Gladys Grenot's lab that she took 145, I know for AV9, a couple less samples for the other serotypes, and ran ELISpots to look at interferon gamma positive T cells against different capsids. She also performed IgG binding assay to look at patients that were either positive or negative on that assay. And something that I found really surprising was among the IgG negative patients, there were a fair amount of these are not patients, these are donors again, donors that were actually interferon gamma positive. And so this is suggesting that there are potentially patients that are seeing AAV capsid that are non-naive, but that are not showing up, at least on this IgG binding assay, as being positive. And one thing that additionally was interesting is when they tried to figure out whether this was a CD4 or a CD8 response, they actually saw that when they depleted the CD4 T cells, that they had a strong interferon reaction where they did not see that with the CD8. So this is suggestive that it's a predominant of a CD8 response. However, PBMCs do contain other cells that can make interferon other than when you deplete out the CD4 or the CD8s, there are other cells that are available. So the question is then, how would pre-existing immunity influence the T cell response to AAV gene therapy? I think certainly this is an important question. If we deplete all the B cells and now we can redose patients, for example, are we going to see if these patients have already been exposed to AAV and they might have a T cell response, are we going to see even stronger cytotoxic T cell response? Juliette mentioned that a lot of the models that we've been doing all this preclinical work is in non-human primates and in mice. And mice and non-human primates do not really recapitulate the CD8 T cell responses that we have observed in clinical trials. So these are really hard models for us to get an idea of what's going on as far as the T cells are involved. I bring up this trial because obviously human data is very helpful, and multiple people have shown this slide, but one thing that I was really interested in is looking at transient tramamide or elevated liver enzymes in these patients, either with patients that had NAbs versus didn't have NAbs. And one thing I personally found a little bit surprising was that the patients that had NAbs actually had less indication. So this isn't an actual measure of T cell responses, but it is an indication that there is a capsid-specific T cell response going on. So at least in this particular trial, it didn't seem to have effect on the T cell response, and if anything, it was slightly decreased. And so we know that having a cytotoxic T cell response can be an issue for long-term transgene expression. That is the reason that we do a number of broad immunosuppression, but are there ways that we can modulate T cell responses so that they can be beneficial? Certainly in the alpha-1 antitrypsin trial, we saw that there was a real beneficial benefit to inducing T regulatory cells. And so a number of groups have been interested in trying to induce T regulatory responses. Certainly in mice, this is quite easy to do in the liver using AAV. But there are other groups that have used mTOR, for example, to try to induce T regulatory responses, which is a lipid nanoparticle that contains rapamycin. However, my lab has been interested in trying to engineer T-reg responses instead of trying to induce them, though we do have a number of experiments where we're also trying to induce T-reg responses. So just to remind everyone that when we do get T cell responses that are specific against AAV capsid, it can result in loss of transgenes. So the general idea was that if we created a CAR Treg, so if we borrowed technology from cancer and used a chimeric antigen receptor that actually recognizes the AAV capsid and turned that T cell into a Treg instead of an effector T cell, could we then suppress immune responses and allow for sustained transgene expression? And so this is the design of our CAR Tregs. So we have a AAV-specific scFv. We've tested this against a number of AAV capsids. I don't have the data here, but it's in the paper. But we looked at AAV2, AAV5, RH32, 33, AAV8, AAV9. So a lot of the common AAV capsids, and they all worked. This is actually a third-generation CAR. We are now using a second-generation CAR. And we've included into our CAR construct FOXP3 because this is the important transcription regulator that creates the Treg phenotype. And basically, it's really important for us that A, the cells don't turn off FOXP3 and lose the Treg phenotype, so it's expressed off of our promoter and not off its endogenous promoter, as well as it allows us to make, Tregs are very low population, and it allows us to manufacture more CAR Tregs. And so our initial experiments, what we did is, I just said that mice are not a really great model of looking at capsid-specific immune responses, but we do have the ability to use this one virus, which has already been brought up a couple times today, which is RH3233, which does induce a B and T cell response in animals. And so we use an alpha-1 antitrypsin as a reporter gene in these animals, and we injected intramuscularly in mice the RH3233, and then 1 week later, we either injected PBS, our CAR Tregs, or we used polyclonal CAR Tregs. And what you'll see at the bottom here is in blue, when you deliver PBS, we initially get expression of alpha-1, and then it drops off by about 5 weeks, and when we go all the way out to 26 weeks, we see very low levels of expression. However, when we deliver our AAV CAR Tregs, we see that we get sustained long-term transgene expression over the course of the experiment. And so one thing that we evaluated, and it's important to this crowd certainly, is we looked at antibodies against the capsid, and if anything, we saw higher antibodies against the capsid using our AAV CAR Tregs. However, we were delivering the AAV CAR Tregs 1 week after the AAV delivery, which probably limits their ability to suppress any immune responses. And there are other papers using AAV CAR Treg, or sorry, not AAV CAR Tregs. This is a hemophilia CAR Treg that was designed for protein replacement therapy, and they saw that they could dampen the B cell responses. However, our CAR Tregs were producing a number of important suppressive cytokines like IL-10 and TGF-beta. And when we looked in the actual tissue, we can see that we completely lost expression in the muscle of the animals that just received PBS, where in the animals that got the CAR Tregs, we were still able to see good expression, and that correlated as well with the vector genomes in the tissue. So the next question we wanted to ask was, can CAR Tregs suppress the transgene immune response? One of the really unique things about Tregs in general is that they can be broadly suppressive through bystander suppression, which is locally they're producing these important suppressive cytokines like TGF-beta and IL-10, so they can suppress other cells in the area. They can also induce through infectious tolerance other tolerogenic cells, and they might have different specificities, so you can actually suppress other antigens. So even though our AAV CAR Treg is directed against the AAV capsid, we wanted to see if it could actually suppress the transgene immune response. And so here we use AAV1, which wouldn't cause really a T cell response in the mice, but we put in a really strong antigen, which is OVA in these animals. And so again, in the blue lines where the animals got AAV1 OVA, but PBS, we see that there's a low level of OVA expression. And when we look at the histology in these animals, we see robust cellular infiltration and myositis. So there is severe inflammation in the muscles of these animals. However, when we look at the animals that receive the AAV CAR Tregs, we see much higher levels of expression that it's sustained out to 16 weeks. And when we look at the histology of these animals, there is significantly less cellular infiltration in these animals, suggesting that not only are we getting expression of this immunogenic protein, but it's also being tolerized, and we're not getting this robust immune response. So just to overall conclude from all of the topics of the talk, we know that T cell responses against capsid or transgene, which I didn't really talk about, can result in loss of transgene, elevated transaminases, and lead to issues in the liver. However, T cell responses can also be beneficial, and inducing Tregs can lead to long-term stable transgene expression. Little is known and studied about cellular immunity to AAV, and I think that's something that we need to consider as we're considering treating patients and coming up with strategies to overcome B cells, we also have to think about what are the T cells going to do. And then potentially inducing Tregs or even engineering them can be useful in modulating immune responses. And finally, I showed that our AAV CAR Tregs that we developed can modulate immune responses, resulting in stable transgene expression to both immunogenic capsids and transgenes. And with that, I'd just like to thank my team. A lot of this work was done by Moda Hare in the lab, who's now a graduate student. And thank you for your time.
Kathy?
Hi, my question's for Alison. Given that the AV capsid is recognized by the CAR, I mean, presumably that means there's plenty of capsid around that's activating these T cells. Have you explored this? And how do you think these CAR Tregs are becoming activated if the capsid is inside the cells?
So that's a really good question. So we actually took the antibody that we used to make the CAR and stained 6 months after injection, and we were able to detect staining on the surface of the cells with the antibody. So we know that AV capsids
sticky in muscle cells?
In muscle, yeah. So we know that capsid's sticking around for a long time. However, I think that probably what we are doing is that the AV CAR Tregs are inducing other tolerogenic cells that are prolonging. So I don't know that we're actually getting the AV CARs recognizing capsid forever, or more that we're then inducing other Tregs that are going to continue to recognize antigens that can be presented in, because the capsid is going to stick around inside the cell and continue to be presented.
So to that point, you mentioned you transferred the CAR T cells 1 week after treatment, but have you looked transferring other times? Like, is there some point where they're no longer effective?
So we haven't gone out much further than 1 week. Let me think about that if we have. We have tried earlier because we're interested in trying to kind of blunt the antibody responses. But we don't know, I guess, where we obviously, in order for these cells to survive in vivo, we need to have a high antigen load, which is why we kind of deliver them in a time period when we think they'll be able to see a lot of antigen.
Okay, great, thank you.
Yeah.
So I have a question for Ben, and I don't know if you can answer it, but well, no, I mean, you'll see. I mean, I find it surprising that you could actually evolve an AAV capsid that is resistant to a broad panel of neutralizing antibodies. And I'm wondering, so it's manufacturable, it still hits the transferrin receptor. I mean, I guess you can't tell us how conserved is it compared to AAV9, or how not conserved is it?
Well,
like, is it 80% identical? Is it 60%? Is it?
It's more identical than that. I can't say too much. You're right that I can't talk about that yet. But I think the approach we're taking is different than what's been tried in the past. I'll say that. And I can't really comment on how many mutations we're putting in exactly, but it's not getting us to something that is only 80% similar to AV9.
So in other words, if you had structural data on this capsid.
I mean, there's AlphaFold and ways to have some structural data, but we don't have actual cryo-EM or anything.
But
yet.
if you did, just from AlphaFold, right?
Yeah.
Does it look like an AAV capsid?
Oh, it looks exactly like an AAV.
Okay.
You would see it and you would think that's an AAV capsid. Yeah.
So just to follow up on that, presumably they're manufacturable, but how much so? Like, do you take a hit with these mutations?
Some of them we take a little bit of a hit, others we don't. I mean, we look at that very carefully. Even at the library stage, we can get a pretty good sense which ones will be more difficult than others. And we so far, the capsids that we are focusing on are ones that are within twofold of their parental capsids. So they're in terms of their lysate yields, and so we think they're going to be very manufacturable.
Okay.
And those parents are within that of AV9 too. So these are not that different from AV9, which is one of the better producing capsids.
Question for Alison, really. In terms of the induction of Tregs, we've seen some nice data with CTLA-4 at this meeting. Are there other near-term approaches that could be deployed in the clinic? I guess thinking particularly of anti-CD3, which has recently been approved for pre-diabetic type 1s. Are there other interventions that might be appropriate in this setting of AAV dosing to enhance Treg generation?
Yeah, so there are a number. Certainly there's some work with IL-2 muteins that will specifically bind CD25. And so there's an antibodies that will kind of induce T regulatory development. And so I know, we have a number of colleagues and people who are working on kind of strategies to, at the same time you deliver AAV, to induce or kind of shift toward a T regulatory response.
I guess one more, and then we'll turn it over.
Another question for you, Alison. Is it futuristic to think about engineering Treg cards against or for transgenes, for specific transgenes such as dystrophin, for instance?
Yeah, so I actually showed that someone has developed one against one of the factors for hemophilia. So certainly people have considered engineering against transgenes as well. And like I said, one of the great things about Tregs is this, bystander suppression, that if you can get them in the right location, that they should suppress. So I think there's a growing field now of people engineering CAR Tregs for a number of indications, certainly autoimmune diseases and neuroinflammatory diseases, but also against transgenes. Are we?
I think we're.
Just a rapid question about the capsid engineering.
Yeah.
So I was wondering, what's the trade-off between biodistribution and prevention of the NAB? Because it's clear that you can still improve, right, the NAB resistance of your capsid. But I was wondering how much you have tried to push actually the NAB prevention, and if in that case you're going to risk to lose the biodistribution. It's related to, you're going to change massively the structure, and in the end you will. So I was wondering whether there is a possibility of doing a double directed evolution.
Yeah, I mean, we are doing these screens in a way where we are very, we work really hard, actually, or Simone works very hard, and others in my lab work very hard to check all of these things from the lab. And so we're really screening many, many, many thousands of different capsids in a quantitative way to pull out the capsids that are maintained in their ability to engage the receptor that we're trying to keep them engaging, and also have antibody evading characteristics and the ability to still be manufactured. So we think there are certainly cases where we made them lose one of those functions. And so that does happen, but we screen so many that we can really focus on the ones that are still able to do all the things we want. I mean, it's really just a numbers game, and we just carefully do the screens so we can read that out before we spend too much time on the individual characterization. So I'm happy to talk more about it, but I mean, we do it very carefully and quantitatively, as I'll say.
All right. So due to the challenges of plane travel in 2024, we've had to make some slight adjustments. And so Dr. Wilson is going to be joining us by Zoom. And so I'm going to ask you guys to stay in your seats for just a couple minutes while we do a quick sound check. And Dr. Marks, go ahead.